the physiological unknowns are coming in very piecemeal, but I have more physiological unknowns than one. I think I'll post both of those when I post the lab exam scores. So for those of you that haven't turned those in yet, just recognize when that 100 points goes into the progress report, it's going to really knock down some percentages.
For both pumps, right?
Yeah,
not semester project yet.
Yeah, not the semester project yet, no, but the two physiological unknowns, because some people turn them in and they'd like to know, how that's affected them.
Yes.
So I'll try to get those.
Agreed.
Okay, so did everybody get this study guide for laboratory exam 2?
Yes.
So what I want to do is just start going through this, and then when it gets to the part where I know I did finish it, I have some, actually I made up some material for you guys to look at for the water test because we didn't actually get to see it. So I set some data up, and I'll pass those out so we can go through that part, and then I'll go over the composition of blood. And you look at the white cells. Well, some people look at the white cells, some people don't. Okay, so laboratory exam 2 for you guys starts with the exercise on determination of microbial number. I believe that the other lab sections, that was on their laboratory exam 1. But for you guys, it's on your laboratory exam 2. So there's one example question out here that has to do with bottles of water. Remember that what you're looking at is the quantitative plating method. It's also called the viable cell count. A question that you're very likely to be asked is, why are viable cells the only ones that can be counted? It's because viable cells are the only ones that will grow and form colonies that you can see. If you tell me viable cells are the only ones you can see, I'm going to say that's not right. You can't see cells. Whether they're alive or dead, it doesn't matter. You can only see colonies. They have to grow and form colonies. You need to know what a serial dilution is, and you need to know how to calculate the degree of dilution. So we'll probably need to go through that in detail. I don't want to do it right this second, but I'll come back to it after people have had a chance to look. Again, the most important thing is to recognize that when you're doing a serial dilution, you're doing a dilution that involves a series of containers. So you need to be able to figure out how to determine the degree of dilution in each bottle. You have an individual dilution factor. You need to know what those are. Then to determine the total dilution factor, you just add the exponents together. So you want to make sure you get your individual dilution factor in the form of exponents. In other words, 10 to the minus 1, 10 to the minus 2, 10 to the minus 3. Don't leave it in fractions.
We do dilutions for the virus one and for this. Are they the same or are they different?
They're the same, except the virus one is always 1 in 10. It's always 1 in 10. Okay. Whereas the water one, I mean, when we did the serial dilution for the bacterial count, some of them were 1 to 100 and some were 1 to 1,000.
Okay.
So it's a degree of dilution.
Thank you.
The other thing you need to know how to do with that determination of numbers is be able to count the colonies, multiply times the dilution factor. So you have dilution in the water, and then if you take 1 mil and put it on a plate, that's not a dilution, because you're always wanting to know how many cells are present in 1 mil. But if you transfer 1/10 of a mil to a plate, that's another 1 in 10 dilution, or 10 to the minus 1. You have to add that into your series. And then you count the number of colonies, multiply times the dilution factor, make sure that your final number is in correct scientific notation, only one number to the left of the decimal point, and that your exponent goes up, not down, when you move the decimal point over one place to the left. The next exercise had to do with the selective and differential media usage and your replica plating. I didn't find any real nice plates that have MacConkey's, growth on MacConkey's or growth on T7, so I put a T7 plate out there that doesn't have any growth. By Thursday, I'll have some good plates. But you do need to know what a positive and what a negative looks like on those 3 different media: MacConkey's, T7, and mannitol salt. We did not use EMB for physiological unknown 1. So as far as the replica plating, although we used EMB, that's not one of the plates I'm going to be asking you about. So you need to know about MacConkey's and T7. Both of those are differential on the basis of lactose fermentation. They are both selected for gram-negative. Mannitol salt is selected for halophiles because it contains 7.5% salt, and it will show you if the organisms can ferment mannitol or not. McConkey's and T7, they are going to be fermenting lactose, and mannitol salt, they're fermenting mannitol, and you need to know what fermentation looks like and what non-fermentation looks like. There's plates, there's pictures of the replica plates on the website, so you can go through and review that pretty easily. Beyond that, you need to know what the replica plate device looks like and the significance of replica plating. We were able to transfer 5 colonies all at once. That saves time, it saves media, it saves money. Also know the function of the nutrient auger plate at the end. That was a control. If you had all 5 colony types growing on that last plate, then you know that you transferred all 5 colony types through all of the media. That's what we used that for. Then we started a series of tests for the physiological characteristics of bacteria. That came in 3 different sections. You don't have to worry about the sections, but you need to be able to recognize and interpret the results of those biochemical tests. There's a lot of detail here. So there's a bunch of different pH indicators that you need to know. Phenol red, bromophenol blue, bromocresol purple. You need to know how those things work, what kind of color changes. You need to know what's being tested for in each one of those tests. The OF, it's oxidation fermentation, but it's telling you what kind of metabolism the organism uses. Are they fermentative or are they respiratory, which means they're using an oxidative metabolism. The carbohydrate deeps, we're using them to determine can the organisms ferment various different types of carbohydrate. TSI, primarily we're using that to show can they form hydrogen sulfide from the metabolism of sulfur-containing amino acids, but it'll show you sugar fermentation. The pH indicator in TSI is phenol red. SIM, 3 tests, sulfur, indole, motility. You're testing for hydrogen sulfide production, indole production, and motility, and you need to know what those results look like. Citrate utilization, urea hydrolysis. You need to know the names of the enzymes involved, citrate permease and urease. You need to know the pH indicators. In citrate, it's bromophenol blue. In urea, it's phenol red. You need to know what positive and negative results look like. For the amino acid decarboxylation, you need to know why are there 2 tubes? What's the function of the control? Those media start out purple, and purple is the neutral color. It's also the alkaline color. So you're not going to be able to see the formation of an alkaline amine unless you can show that the organisms are capable of fermenting and producing acid that would turn the medium yellow. That's what the control tube is for. It says they can ferment glucose, there's nothing else in there. If they can ferment glucose, they'll produce acid and the medium will turn yellow. That means then if your lysine tube is purple, it's purple because they made an amine, and the amines are alkaline. And that's why the bromocresol purple is purple there. Oh my goodness, phone is dinging. For the MRVP, you need to know the MR, the methyl red test, is testing for large quantities of acid. The VP test, the Rhodes-Proskauer test, is testing for acetoin, and not vice versa. Don't tell me the MRVP, that the VP test is testing for acid production. It isn't. It's testing for acetoin, and acetoin is neutral. Beyond that, you need to know kind of basically how those are done, that the MR test requires methyl red as an indicator. VP is requiring Barrett's reagents A and B, and then you have to shake the medium. Sorry, I have no idea what that's about. I'll turn it off, sorry. Okay, so catalase test. Know what a positive catalase test looks like. Bubbles. It's a test where you test bacteria by exposing them to 3% hydrogen peroxide. If they bubble, they're catalase positive. Oxidase test, you rub a sample of the organism into filter paper. If they produce cytochrome C oxidase, the filter paper turns purple. Esculin hydrolysis, if they can hydrolyze esculin, the medium turns from kind of a gray tan to black. We're not looking at very much detail on that one. Coagulase test, you have rabbit plasma. You put the organisms into rabbit plasma. Don't shake those tubes, by the way. You just turn them over, and if the plasma doesn't move, then it's coagulated. It's solid. That means coagulate positive. If it's liquid, it's coagulates negative. And then you need to know about hemolysis reactions, alpha, beta, gamma hemolysis. The tests that you do not need to know for this exam are the nitrate reduction. Some people did that, but I'm not going to ask you about it because not everybody did. We didn't do starch hydrolysis as a group. Some people did, some people didn't. And we didn't do gelatin hydrolysis as a group. They're in the lab manual, but you don't need to know those. As far as hydrogen sulfide production, you need to know that hydrogen sulfide comes from the catabolism of sulfur-containing amino acids. People want to tell me H2S is produced when the organisms catabolize sulfur. I was like, no. They're catabolizing sulfur-containing amino acids. I think there's only 2 of them that have sulfur in them. They have to take those things apart, and they produce hydrogen sulfide. It reacts with iron in the medium, which forms iron sulfide, and that's black. That black precipitate is in the bottom of a TSI tube or in SIM. It's the same black color. By the way, that's not the black color that's in the Escalent tube. That's not H2S. Yeah.
So one more time, hydrogen sulfide comes from sulfur-containing amino acids?
Yes. Yes. Okay, so then we did the application of the PCR and the identification of unknowns. So you need to know basically information that had to do with the PCR in the physiological unknown 2 report form. What is the polymerase chain reaction? How does it work? What is it basically showing us? It is DNA replication happening in vitro. So it's DNA reproduction or amplification. It involves Taq polymerase. The Taq stands for Thermus aquaticus. So that's a enzyme that's coming from hyperthermophilic bacteria, very heat resistant. Why does that matter? Because during the PCR, when this little machine is running, this thing is cranking up the temperature to 94 degrees over and over and over again. It runs, well, the last time I had this thing set, it was running 37 cycles. The last time I looked at it, it was running 50 cycles. That means 50 times it's cranking the temperature up to 94. It starts off at the beginning for 4 minutes at 94 degrees. That's really hot. Most proteins are denatured at that temperature. So the Taq polymerase isn't, and that's the significant feature of the PCR. If you tried to use a protein that wasn't from a hyperthermophile, it would probably be denatured and the PCR wouldn't work. So where did this thing usually come? Where did it originally come from? Again, Thermus aquaticus, lived in the hot springs in Yellowstone National Park. You need to know what primers are, which primers we use, how primers bind. So there's a question over here that has a diagram of 2 strands of DNA. And the strands are separated, and the question has to do with where would primers bind? You will likely see this kind of thing on the test, where you're going to see a diagram. You have the denatured strands of DNA. One reads 3' to 5'. The other one reads in the opposite direction, 3' to 5', because the DNA strands are anti-parallel. Now the question is, where do you put primers? The primers go at the ends of the gene we want to amplify. If this is our 16S RNA gene, we know that one of the functions of primers is to delineate, to bind at the alternate ends of that so that they determine what gene is going to be amplified. But where can we add nucleotides? 5' end or 3' end? 3'. Therefore, you have to look at this and say, okay, where can I put this? My end here has to be opposite the letter or the number that's there, so this has to be 5'. This thing can be here. Here's 3'. That can then be the free 3' end that Taq polymerase can add nucleotides to. That has to be toward the middle. If you have a 3 here, this has to be your 5. These have to be opposite one another, or excuse me, that's not opposite. Complementary. They have to be complementary. So if this one's here, then the other one has to be over here. Again, so that your 3' is toward the middle. You can build this way on a 3'. If you were to put this down here and say, okay, I'm going to put it in this position. Here's my 3', here's my 5. That's not going to work. You can't add nucleotides to a free 5' end. You can't. The polymerase can't. The polymerase can only stick nucleotides to a free 3' end, which is one of the functions of the primers to make sure that that end is there. Make sense? So the primers provide that free 3' end, They make the DNA a duplex. This thing has to be a duplex. Even this little short amount is enough for Taq polymerase to bind now. DNA polymerase can only bind double-stranded DNA. So it's going to determine what gene gets amplified. It's going to provide that 3' end. It's going to make the DNA a duplex. It also determines the temperature, which is not a big concern to you guys, but if you're trying to design primers, it's a big concern, because if the primers aren't the right temperature, then your anneal temperature isn't right, and the PCR doesn't work. Okay. The other thing is what gene was targeted. Again, 16S rRNA gene. Why? Because all prokaryotic cells have it, and because it's highly conserved over evolutionary time. It hasn't changed very much, so it's a good indication of evolutionary progress and relatedness between organisms. The next exercise with genomics, proteomics, bioinformatics. You need to know what those terms stand for, what they mean. We'll be able to define them. Know what the acronyms NCBI and BLAST stand for. It's interesting, the funny things that people make up when they haven't studied. What does NCBI stand for? Or what does BLAST stand for? So NCBI, National Center for Biotechnology Information. BLAST, Basic Local Alignment Search Tool. You need to know those. It says you need to be able to copy a nucleotide sequence from the Biosoy4 webpage and access NCBI and get the BLAST algorithm. I am not going to ask you to do that during the exam, because if we tried to do that, chances are NCBI would just be shut and we wouldn't be able to use it. Everybody would be in a state of high frustration. So you're just going to see a paper image, and then you have to be able to look at it just like you would if you pulled up the BLAST result page. You need to be able to look at the page that you go, if you click on the accession number, you go where you see your taxonomic lineage and that kind of thing. So it's just going to be on paper. And then we did the mini screen, rapid isolation of plasmid DNA. Actually, I think we did the digestion first, but anyway, we did the mini screen. So you need to know what plasmids are. You need to know why we extract them from bacteria. The plasmids we were working with, PUC19, PGEM, PGLOW, they're all cloning vectors, and they're also expression vectors. You need to know what those terms mean. So cloning vectors, that means they can carry DNA into a cell and reproduce it there. Expression vectors means they can express the DNA they're carrying. They can express the genes. Those 3 plasmids all express beta-lactamase, the marker genes that encode the enzyme beta-lactamase, so that they can be resistant to ampicillin. And what else do we need to know about them? Well, some other stuff, but that has to do with the transformation exercise we'll get to in a minute. There was the restriction endonuclease, digestion of DNA, and RIFLEP. So you need to know about restriction endonucleases, what they are. So they're enzymes normally produced by bacteria, probably made bacteria resistant to viruses, and they are named according to the bacteria that they came from. Typically something like EcoR1 tells you the genus name, the 2 first letters of the species name, a strain identification, and a number that tells you whether they were the first, second, third, fourth enzyme found. So you need to know about that naming system. You don't need to know about neoschisomers and whatever the other schisomers, isoschisomers, never mind. Need to know what a recognition sequence is. So a recognition sequence is where the enzyme binds. They typically cut within it. And if they cut, what kind of termini are they going to produce? They're going to make blunt cuts, or are they going to produce cohesive termini? And if they're cohesive termini, how many bases long are they? The example that I gave you was the GAA-TTC, which was the recognition sequence for EcoR1. It's in the lab manual. You need to know that one. And then you need to know what RFLP stands for, restriction fragment length polymorphism. So if you cut DNA with a restriction enzyme, you get a bunch of pieces. We cut with the ALIN1 enzyme. The recognition sequence is AGCT, cuts between the G and the C. You've got to do that and make your RIFLET patterns on paper. Some of you still haven't turned that in, by the way. That's 10 points. Turn in those RIFLET patterns. And yes, it needs to be your physiological unknown too, as opposed to somebody else's randomly selected from I don't know where. It has to be. If you don't know what your number is, ask me. But I want those turned in. They're worth 10 points. Come on. There's some homeworks in there. Yeah, there's DNA homework needs to be turned in also. Anyway, so RIFLET patterns are basically DNA fingerprints. They're just patterns you create when you cut DNA into a bunch of fragments and then run it in a gel. The fragments sort out, smaller fragments move faster through the gel, and you get these patterns. Which means you also need to know about gel electrophoresis. That's the next section. So you need to know what the equipment looks like, how the gel goes in. So I have a box out here, and there's a question about, is the gel in the box correctly? You have to look at it and decide, is it in there right? If you turn the power on and there's buffer solution in there, the DNA is going to move. The question is it going to move into the gel, which is what you want it to do, or is it going to go out the short end and into the buffer solution? If it does that, you've lost it. So it's important that you know how the gel goes in there. Let's see, you need to know, so DNA has a slight negative charge, so it's traveling toward the positive pole, toward the anode. You need to be able to recognize the sizes of our plasmids by the pattern they look, how they look in a gel. You have to be able to recognize the lambda DNA cut with Hindi 3. So if you have a gel, bacteriophage lambda is always 1, 2, 3, 4, 1, 2, 1. These are very close together. This one may not show. This bottom one is approximately 4,000 base pairs, or 4 kb. These two are approximately 2,000 base pairs, or 2 kb. This is 564. So you can use that then to measure and get an idea of size-wise how big your plasmids are. PUC19 will line up either with the 2,000 or slightly below that. So if you get a band here, it's PUC19. P glow will line up approximately here, even though it's 5,000. It looks smaller because it's supercoiled. And PGM is somewhere in between. And then our PCR product would be right in here someplace. So you need to be able to recognize where those things are in the gel. Okay. So then we have the calcium chloride procedure for making competent cells. You need to know what competent cells are. They're basically cells that are very ready to take up DNA. My little cartoon cell was up there saying, I'm so ready. They're just more capable of taking up the DNA. To make them competent, we grew them to mid-log phase and we treated them with ice-cold calcium chloride. The calcium is a positively charged particle. It's going to make the surface of the cell slightly positive, so the negatively charged DNA will get close and they can take it in. At least that's the story. There's enzymes involved too, which you don't need to worry about. Anyway, so we had to make our competent cells. Don't worry about all the procedures that we went through to do that. And then we did transformation. So you need to know what transformation is. Transfer of DNA from dead donors to live recipients. And you need to be able to look at the data that we collected and explain some things. For example, why do you have isolated colonies on the TSA amp plates and you have lawns on TSA? On the lawns, everything grows. TSA, all the cells can grow. But on TSA amp, only transformed cells can grow, and the transformed cells make up about 10% of the original population. We didn't actually calculate that, but it's documented about 10%, which is not very much. So you get isolated colonies. The other thing you need to know about is the arabinose operon that has the green fluorescent protein gene associated with it. You need to know that arabinose is the inducer, that the repressor that is encoded by the ARC gene is constitutive and active alone. So you have to have an inducer in order to express the green fluorescent protein, and the inducer is arabinose. And then we have our introduction to viruses. We have phage typing, and we had the isolation of colophage from the environment. You need to know about phage typing. In other words, be able to interpret the data. As I said, the names will be changed on the test. They won't be E. coli C and E. coli B. They'll be E. coli with some other crazy name, and the viruses also have different names. But there's an example of phage typing plates. You need to know what phage typing is, using known types of bacteriophage to identify unknown types of bacteria, and be able to interpret the data. It's not terribly difficult to do. And then as far as the isolation of colophage from the environment, that's another serial dilution situation where we had to take a sample from we stabbed plaques and collected a bunch of bacteriophage in a little tiny tube, then did a serial dilution, and then plated out those different dilutions to see how many phage particles were in 1 ml of the KCl broth when we put them from the stabbed plaques. Every one of the steps of the serial dilution, each individual dilution factor is 1/10, or 10 to the minus 1. And again, to figure out your total dilution factor, add the exponents, 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, just add them up. You guys can add 1 and 1 is 2, 2 and 1 is 3, 3 and 1 is 4. Just remember that when you go from a tube to a plate, if the amount transferred is 100 microliters, which it was, that's 1/10 of a mil. That's another 1 in 10 dilution, or 10 to the minus 1. Okay. Then we had the bacteriophage reproduction and plaque formation exercise. You need to be able to read the graph. I'm not going to have you make the graph. You need to be able to read the graph. So there's an example graph out there, and it has red lines on it. You need to be able to see where those lines come to the left-hand edge where you're reading the log scale, and be able to read it so that you can get the numbers. You need the log, you need your initial number, and then your maximum number of virus particles. The initial number is infected cells, the final is free virions, which doesn't matter too much, but so you need to be able to look at the data that's on the graph, determine burst time, or latent period. How long does it take for the virus to complete its life cycle? So you look for the big increase in number. You've got a bunch of low points and then suddenly you have a bunch of high points. What's the time interval when that jump occurred? You can't get closer than 10 minutes. You can say it's between 30 and 40 minutes, or it's between 40 and 50 minutes, whatever the data shows you. And then you also have to be able to calculate burst size. How many virions are released per infected cell? So you have to know how many infected cells have you got, and then how many free virions. Divide the number of infected cells into the number of free virions, and that's how you get the number of free virions released per infected cell. Anyway. So then we did the bacteriological examination of water, only you didn't. So let's look at that. Just let me get my data out here. I'll go through this as quickly and painlessly as possible.
It's going good.
Except they stink. Sorry about that.
And then
Sorry, don't drop that.
are we still?
No, we still have
I've been kind of watching these all afternoon.
Because she's going to take a different topic, so it's going to stop it.
And other things.
No, she's going to a topic on here.
Yeah.
We need to take this too.
Yeah, based off that review, I feel pretty solid. This does.
This review?
I've also ran through this practical exams like 6 times.
What is it?
Son, did you turn this in?
She left me down for
You
that
got me
one.
an
I was like,
18 and a half. 18. Yeah, you're
so it's okay.
Okay, so I wasn't really sure how to present this information, and ultimately I decided, well, the best thing to do is give you some example water samples to look at. That's what these are. Only when I came over here to set this up last night at about 8:30, there was no double-strength phenyl red lactose broth. So then I had to make double-strength phenyl red lactose broth, which had to be autoclaved because there's germ tubes in these things and they won't sink if you don't autoclave them. So this way, at least each group has something to look at.
Come on.
I thought it was broken on the bottom.
of these for every group, 2 of them. So maybe these will have to move around, but real quick, we'll go through the bacteriological examination of water.
She will.
Okay, so right off the bat, if I were to ask you, why do we care about examining water for bacteriological content? What do you do with water? You drink it. A lot of us drink it right out of the tap at home. We use it to wash our clothes. We use it to wash our hands. We use it to brush our teeth. It'd be nice if it was clean. In this country, we kind of take it for granted that it's clean. But in many countries, it is not. And you don't take it for granted. Nevertheless, that's what this is about. Why? Because we use water for drinking. We drink it, we wash in it, etc. We want it to be clean. What's significant is this is the bacteriological examination of water. We're not talking about analyzing water for lead content. We're not in Michigan. We're not concerned about that, although certainly people need to be, because the pipes that are carrying water are potentially making it toxic. I often wonder about how much stuff is coming out of PVC, because most of our water is now being carried in PVC pipes. That stuff is being coated by biofilms on the inside. I don't know what the microbes are doing with that stuff. Supposedly they can't break it down. My thought is always, not yet. When they learn to, oh my word, we're in big trouble. But anyway, when we're looking at the bacterial content, we're interested in what we call an indicator species. And although there have been proposals for a variety of things that might be considered, typically the indicator species is Escherichia coli. My thought is probably this occurred because Escherichia coli was identified very early on, and because people recognized this thing lives in the gut. So as an indicator, if we said why do we use it? One, lives in the gut. Lives in the gut of humans and other animals. It does not live in rotting vegetation. If it's in water, that means that water is receiving gut contamination, fecal contamination. And that's something we're concerned about. The other thing about E. coli is it's easy to grow in vitro. Another organism type that you should recognize the name of, Giardia, also lives in the gut. It doesn't live in association with rotting vegetation, it only lives in the gut. If it's in water, it's an indication of fecal contamination. But Giardia will not grow in vitro, period. As far as I know, they've never developed a medium that will grow that particular organism. Whereas E. coli, they're really easy to grow in vitro. The reason we use it is it's easy to test for. And this is probably because, as I said, it was discovered quite a long time ago. Lots of people have been working with Escherichia coli, and they've developed a lot of different media that allow it to be recognized and identified and tested for, all in one step. Well, so what we're using for our bacteriological examination is referred to as standard methods. The type of testing that is typically run if you're looking at well water samples or river water samples. Different agencies are constantly testing water. I know in Auburn, diamond well drilling, they started testing water because they were testing water from the wells that they were putting in. But more recently, they've taken over testing, I believe, for the city of Auburn. They do all of their water testing. So they're checking to find out, is water You know, is it clean? That's what they wanna know. So the standard methods, there's 3 parts. The first part is called a presumptive test. And a presumptive test as we're running it is a double strength phenol red lactose peroxide. What does this mean if it's double strength? I mean, what would you assume it? We're talking media here. So if it's a double strength medium, yeah, it has twice the concentration of nutrient that you really need to grow the organisms. This medium contains peptone, but it also has lactose in it. We know lactose is a disaccharide, it's a carbohydrate. We know that phenol red is a pH indicator. So this medium has a double strength of the pH indicator and the nutrient needed to grow organisms. Consequently, what you do is dilute this with water. So the water that you want to test goes into a tube that contains the double strength phenol red lactose broth. This is what it looks like. It's just a clear red liquid with a Durham tube in it. When you put the water in, you're supposed to put water in to double the volume. And that's what you get if your presumptive test is negative. On account of that didn't have any time to grow. But it was probably going to be negative anyway because it's tap water. Well, it's DI water. I don't know. I haven't tested this stuff. Typically, if you test chlorinated tap water, this is what you get. This is a negative presumptive test. Phenol red, our pH indicator, no change in color, no gas in the Durham tube, no cloudiness. So that's a negative presumptive test. These tubes that you have contain water from various sources. So this is pond. This is the same water that we tested when we did our water sample, where we did the effective temperature on water, the cell viability. So notice the degree of cloudiness. So that cloudiness, that turbidity is one thing that tells us this is a positive test. The other thing that tells us this is a positive test is the color change. The medium starts out red. If we've got yellow, what does that tell us? Acid. Phenol red is the indicator. Acid has been produced. Acid was produced from the fermentation of what? Lactose. So Escherichia coli can ferment lactose. These tubes all have a good degree of cloudiness. We see bubbles up the top, that indicates gas production. Some of the gas is caught in this Durham tube here. This is the bird bath water. There's a little bird bath outside my house. So it has good turbidity. There's gas in the Durham tube. It's yellow. That's a positive presumptive test. Now, ideally, you would do a presumptive test. You would grow this for 24 hours. And then you would use your positive presumptive test to inoculate your EMB. The EMB is the next step in this test series, and that is called the confirmatory test. So you streak EMB, and if you remember from our exercise on replica plating, E. coli colonies on EMB are dark and they have a metallic green sheen, or they have green iridescent color. So we have a lot of plates, a lot of EMB plates that you can look at that are the confirmatory test. Does anybody see the green? I was kind of curious about it because these plates weren't streaked from our water samples. Some of them are E. coli. So I just put E. coli on a plate. There's a little bit of green on that one. They have quite the smell. Anybody else see the green? I did E. coli and Klebsiella, which both E. coli and Klebsiella should give us the metallic green. I also did Enterobacter, Enterobacter will produce colonies that are dark in the center. And then they have a light periphery. They're referred to as fish eyes. So E. coli is dark with metallic green sheen, but if you have growth on a plate, you'd want to look at that plate and say, well, what other kinds of organisms are there? If there are organisms that are usually growing in association with soil and rotting vegetation, they should not be in your well water. If you're testing well water, that means stuff is getting from the surface down into the well, and the well probably needs to be treated. If the only thing that you found in association with the EMB was something like Sporotilus, actually I don't think they grow on EMB, because the EMB is selective. It's a selective medium for gram-negatives. Anyway, those are the only two tests you need to know. Presumptive test, double strength fetal red draw, be able to recognize positive versus negative. Positive is yellow, cloudy, gas in the germ tube. And then a confirmatory test, the EMB, you see dark iridescent colonies, that's positive for E. coli. If you see those kinds of positive results, then that water needs to be tested by an official water testing facility, and if it's a well, it's probably going to need to be treated. I think they dump Clorox down or something along that line. So that's our bacteriological examination of water. Hope that wasn't too painful. Okay, then we did microbial control, where we did effects of temperature. You need to know about the Serratia marcescens or Serratia plymoutica and the production of prodigiosin. When is the red color produced? Only when they're grown at room temp. If they're grown in the incubator, they're white. You need to know about the effects of ultraviolet light. Ultraviolet light is cidal. It kills cells by causing the formation of thymine-thymine dimers, which will disrupt replication and transcription and that kind of thing. So it is cidal. And then the other thing you know about UV radiation, it has very poor penetrating ability. It won't go through the plastic lid of the Petri plate. It won't go through the cardboard. You need to know what antiseptics and disinfectants are, and just kind of the general trend, which was that gram-negative bacteria are more resistant to those chemicals than are gram-positive. That was the only thing we found out about those. Then we did the antimicrobial sensitivity testing, where we got a little bit more specific. You need to know what antimicrobial agents are, what antibiotics are, what a zone of inhibition is, how you measure a zone of inhibition, and look at a chart and determine are organisms sensitive or susceptible, intermediate, or resistant to the various drugs. You need to know where the MIC is, the minimal inhibitory concentration. It's at the outermost edge of the zone of inhibition. You don't have to calculate it, which is the good news. And then if I show you a diagrammatic representation of a simulated blackboard, be able to tell me what drugs are broad spectrum or narrow spectrum from the data that's on that simulated blackboard. And then composition of blood and white cell study. All right, that's another one we didn't do. So I was told to make sure to take these things down before Warren's class tomorrow because he might forget. But for right now, here they are. Maybe you should lay them up. Yeah.
Or take them down for his class
students would like it.
and
Yeah,
up for us.
Actually, I've left stuff like that up and things written on the blackboard up, and you'd be amazed how many people never look at it. Not been so concentrating. Okay, so composition of blood. So, what we need to know about blood, actually it's mostly water, but it has a fluid portion, plasma. So, plasma has mostly water in it, but it has proteins in it. The proteins that we're mostly concerned about in microbiology are the antibodies, because the antibodies are part of our acquired or adaptive immune system. We also know about complement proteins that are in plasma. They're produced by macrophages, but they get released into the plasma. We learned about what complement factors do. So those proteins are significant. also albumin in there, which we don't care about except that we use it for our exercise. We're mostly interested in those proteins. The other thing that's present in plasma are clotting factors, which are significant to physiology. These things are significant to us because we use the rabbit plasma for our coagulase test. So you need to know what a positive coagulase looks like and what a negative coagulase looks like. That is dependent upon the clotting factors that are in plasma. If you take those things out, you have serum. Where did we use serum?
In blood type.
Supposedly. We used it for our hemagglutination reaction, for our blood typing. So serum is plasma minus clotting factors, but typically has a high antibody titer. So we did use serum for our blood typing, anti-A, anti-B, and anti-D serum, supposedly. So, beyond that, we have what are called formed elements. So we have red blood cells, also called erythrocytes. Erythrocyte means red blood cell. And our interest in erythrocytes actually had to do with the epitopes on their surfaces that could be used for hemagglutination. So we were interested in erythrocytes because they're used for blood typing. We have epitopes or antigenic determinants, but what are they called when they're on red blood cells? Agglutinogens. So you need to know about that. You also need to know that antibodies, when you're dealing with blood typing, that the antibodies are agglutinins. So those are two terms that go along with our blood typing exercise, and they have to do with red blood cells. Physiologically, red blood cells are significant because they carry blood gases. Microbiology, we don't care. But yeah, they do that. It's only significant if you're suffering from circulatory shock. You can't get the red blood cells to the tissues, you can't get oxygen to the tissues. Tissues begin to die. That's not good. Then we have white blood cells. These are leukocytes. Leuko means white, cyte means cell. So all white blood cells are leukocytes, and you need to know 5 categories of white blood cells and be able to recognize them. And to recognize that they're all called leukocytes, but some of these individually are called a name that's kind of similar to leukocyte. Don't get confused. Lymphocytes are these guys. Lymphocytes are fairly common. They're not much bigger than red blood cells. They have a very little bit of cytoplasm around a relatively large nucleus. For some reason, students mix up lymphocyte and leukocyte. Remember, leuko means white. Lympho has to do with these cells that are associated with lymphatic tissue, sort of. Anyway, so we have 2 types of white blood cells that are called agranular leukocytes. The lymphocytes, which are the littlest ones, and the monocytes, which are the biggest ones. We talked about monocytes in lecture. They're large phagocytic cells. They have a horseshoe-shaped or kidney-shaped nucleus, supposedly. This is a nice kidney. Most of these are just weird. Monocytes are very pleomorphic. They change their shape because they produce pseudopodia and they creep around. One of the characteristics that you can see is they have a blue cytoplasm, often punctuated by a lot of white spots, bubbles. Those are phagocytic vessels. The 2 that are on the sample here, there's 2 monocytes right together. They both have nice blue cytoplasm with white phagocytic vesicles. Their nuclei are not particularly nice kidneys or horseshoes, but they're pretty big. These are the biggest white blood cells. Anyway, both of these guys that have blue cytoplasm are agranular. Then we have three types of granular leukocytes. Most common are neutrophils, also called polymorphonuclear leukocytes because their nucleus is polymorphic. It can be a wormy looking thing, it can be 2 lobes, 4 lobes, 6 lobes, anything in between, very polymorphic. The neutrophils are called this because their cytoplasm has granules, but the granules are neutral relative to the stains, the dyes that are in the right stain. So they don't pick up the color. Their cytoplasm looks pale lavender. They have granules, you can't see them. The other two granular leukocytes have granules that are visible. Eosinophils, the granules are pink. Typically, eosinophils have a 2-lobe nucleus, but they don't always. Sometimes they're 3. But their granules are definitely pink. And then basophils, the granules are dark. They take up the basic stain, which for our purposes is crystal violet. It's dark purple, and you see these very dark granules. Oftentimes, you can't tell what the nucleus looks like. So we have a nice basophil on here. And it really does make the nucleus hard to see. You just see these purple, dark purple granules on the surface. So one of the things that is mentioned in the lab manual has to do with how numerous these things. There's a mnemonic device, never let monkeys eat bananas. Never is neutrophils. L is the L is lymphocytes. M is monocytes, the monkeys. And then E is eosinophils, and bananas is basophils. That's the order of numerical abundance. So the most abundant are the neutrophils, then the lymphocytes. Supposedly then monocytes, but it depends because if you were having an allergic reaction, you'll have more eosinophils. So monocytes and eosinophils are both relatively low, and those numbers can switch. Basophils are the lowest. They're hard to find, but we have a nice one up here. So you need to know those, be able to recognize them on site. You only need to know what they're doing for lecture. And here you need to know what they look like. They kind of look like the poster. The ones we have up are not too bad. That part. And we even got into the last section here, which has to do with diagnostic immunology. We did that, yeah.
Do we need to know their abundance and
No.
if they're, do we need to know if they're granular or aplanar?
No, you just need to be able to recognize it. No, was all this other information superfluous? Oh, yeah. Sorry. So, serology on the diagnostic immunology. Serology is the study of antibody-antigen interactions in vitro. We study serological tests so we can figure out what's going on inside the body, but also because we use serological tests in diagnostic immunology. The two types that you guys used were precipitation and agglutination. The precipitation test was the Auchterlonie test. There's a little phony looking Auchterlonie plate diagram here with some questions you can answer. You'll see a photograph of an Auchterlonie plate. For the agglutination reaction, we did hemagglutination, that was for blood typing. So that's where you need to know about agglutinogens and agglutinins. You need to be able to interpret the results of a blood typing test. So here's a blood type drawn here, and some questions about blood typing. So you need to know terms like genotype and phenotype and allele and homozygous and heterozygous and who's a universal donor, who's a universal recipient, and why. Who's a universal donor, supposedly?
O negative.
Okay, O negative, because what is donated is packed cells. And the O negative cells don't have A antigens, they don't have B antigens, and they don't have D antigens. So if you only had to worry about ABO blood groups and RH, those O negative cells can't bind anybody's antibodies. They don't have antigens for anybody. Unfortunately, there's more to blood typing than maybe you know. In the lab manual, I got into it a little bit. The other thing is, as far as blood typing, I'm just going to stick to the old system of AO and not pull in the I gene with the A's and the O's as superscripts. To me, that's just confusing. And you'll see it both ways online if you go looking for it. So I'm just leaving it the old way. If you have to learn it for physio, feel free to learn it for physio. The only other thing that's mentioned on here is ELISA, which we didn't do. I mentioned that the ELISA stands for Enzyme Linked Immunosorbent Assay, and the reason it's more sensitive than a precipitation or agglutination reaction is because of the enzyme. Because even a single molecule of enzyme will continue to catalyze reaction, and it will make a reaction visible if you give it enough time. So you can detect a single molecule with an ELISA test. Whereas with precipitation or agglutination, you have to have a lot of molecules, enough to show up in a gel as a precipitate, or enough to pull cells together so you can see it. So the ELISA is a lot more sensitive, but we didn't do one. Okay, you feel sufficiently reviewed? Okay, so the example questions, as I said, are out there. I can set some more example questions up now that we've looked at our water. I know I have some other ones that I might be able to find in my folder, but I kind of ran out of time and space. Anyway, so basically you're free. We will do the lab exam first thing on Thursday because we have to. We do the lab exam first at 5:30. Then we set up, we come in.
And anticipate a lecture afterwards?
Yes. Yeah, because we have to finish. And we're not there
And
then
then Tuesday, will you watch a video during lecture?
during lab. Because during lecture, we're probably going to be going through
Okay.
the
Like
specific
stuff.
diseases.
Oh,
Because
okay.
we still have to do mechanisms of pathogenicity.
Yeah.
And then we have to look at the example etiological agents, which is what we've been studying
Oh,
with our quizzes.
so you know those.
Yeah,
Yeah, you can look at those.
you know those. That'll be a quick review. We'll just look at some pictures and see what those bacteria can do.
Oh, boy.
Oh, yeah.